Good morning, everyone. I'm Linda Tintawi, CEO of the Lustgarten Foundation, and welcome to today's discussion about the future of immunotherapy and what it could mean for treating patients with pancreatic cancer. Research led in large part by today's guest, internationally renowned cancer immunotherapy and translational research expert, Dr. Robert van der Heijden, is expanding into new areas by activating the immune system in a way that has not been done before. This research is all part of Lustgarten-funded science that has been a driving force in every major advancement in pancreatic cancer research since our founding in 1998. Thanks to your support, we fund high-risk, high-reward research focused on early detection and interception, therapeutic development, and personalized medicine. We have a very large audience today, and we encourage you to ask your questions by typing them in the Q&A box down at the bottom of your screen. It's now my great pleasure to welcome Dr. Andrew Rakeman, Lustgarten's Vice President of Research. Good morning, Andy.
Morning, Linda.
Hi, Andy. Patients and advocates from across the United States are keen to learn more about the tremendous advances being made. So I know you and Dr. van der Heijden have a great deal to cover, and we really look forward to this exciting conversation. Thanks to you both.
Thank you, Linda. And I'm very excited to be having this webinar today, and especially excited to be joined by Dr. Bob van der Heijden, who is a professor at the Perelman School of Medicine at University of Pennsylvania and is the director of the Abrahamson Cancer Center there. Dr. van der Heijden has too many participations and credentials and titles to stand here and list, but one of the very important ones is he's also a member of the Lustgarten Translational Advisory Group and a good friend to the foundation, where he helps us to evaluate clinical trial opportunities and look for ways to advance our mission to make pancreatic cancer a curable disease. Dr. van der Heijden has had many scientific and clinical accomplishments. One that he is very well known for is his work deciphering the mechanisms around CD40, which for those who are not immune aficionados is a molecule on immune cells that participates in the activation of T cells. And he's really uncovered the mechanisms to harness that, to use it as a way to turn on the immune system against cancer cells, and has pursued that both in the lab and in the clinic. He's also very well known as leading clinical trials, including two critical platform trials, Prince and Revolution, that I'm sure we'll hear more about. But also, if you dig deep into the literature around immunotherapies for pancreatic cancer, you'll find Dr. van der Heijden's fingerprints in many places, not only around CD40 and Prince and Revolution, but in many trials. So, Bob, thank you so much for being here with us today.
Thanks. Great to be here, Andy, and thanks also to Linda and everyone at the Lustgarten Foundation. It's been a real privilege to be working with you, and I look forward to our conversation.
Yeah, I'm excited for it too. And there's a lot to talk about. Immunotherapies is something that's very topical and very exciting. But before we dig in, I'd like to set the stage a little bit for the audience and get people to understand who it is that they're talking to and what it is that we're going to be talking about. So since I came into the pancreatic cancer world, one of the things I've been really impressed with is the types of researchers and the types of clinicians that get motivated to want to take on such a challenging disease and such a challenging problem. So I was hoping you could give a little bit of your background and kind of what inspired you in both your clinical work and in your research work to focus on pancreatic cancer and focus on immunotherapies.
Well, going way back, I really started my interest in biomedicine as a researcher and as a graduate student. I went to graduate school first, not medical school, and completed my doctorate, and that was in immunology. And then I went to medical school, and I began to understand that there may be a link and a potential for the immune system to react against cancer and actually be a therapy. This is long before the immune revolution, immunorevolution for cancer, which has really changed things for our patients dramatically. We started focusing on pancreatic cancer because of a new laboratory model that was developed here at Penn that allowed us to watch the immune system react against pancreatic cancer. And we suddenly realized that some of the rules of the road that we thought were true for tumor immunology were not true for patients with pancreatic cancer. And of course, being a physician, I was well aware of the amazing clinical need to do something more for patients with pancreatic cancer. So it became an incredible opportunity to bring new insights of the immune system to bear against the problem of pancreatic cancer and what our patients face. We started a center here. This is many years ago at Penn, and at first there were 2 labs who came. That meeting still happens with regularity. Now it's standing room only. The most exciting thing, Andy, is that it's not just faculty at this meeting, it's all the junior scientists, the graduate students, the medical students. There is a huge enthusiasm to take on this hard problem on behalf of our patients. And I've always endorsed that decision I made to specifically study the biology of pancreatic cancer and to see if we can help patients.
That's fantastic, Bob, and it's especially gratifying to hear that not only you took that inspiration when you were first learning about the immune system and first learning about the challenges of cancer, but that you continue to inspire the new folks in your lab and in the other labs at Penn to really take on that mantle. And I think you've touched on a lot of themes that I'm sure we will come back to about the importance of research tools and models and how that opens things up, importance of training and bringing in new ideas. But again, on the mode of setting the stage a little bit, I think if there's the first thing anyone ever learns about the immune system is that it's big and it's complex and it's maybe sometimes confusing. And accordingly, therapies that target the immune system can be complex and maybe at sometimes confusing. So I was hoping you could give us the quick version of immune therapies 101. Like, when we say immune therapies, what is it that we really mean? What types of drugs or treatments or approaches fall under that big banner?
So first of all, it's immune therapy that we're talking about. So these are for patients who've been diagnosed, and can we mobilize the immune system as a therapeutic? And that's really based on our insight that at some level, and it varies tumor to tumor, the immune system finds cancer as foreign. In some cases as foreign as a viral infection, and gets activated. The immune system, the cancer cells understand that. I think we know now that they put up a big front against the immune system. And I always think about that. Why would a cancer do that if it didn't have something really threatening to fear from the immune system? So it took us a while. It took the field a while to understand what would be levers to turn on the immune system, because that's what immune therapy is. To the extent the immune system is trying to get rid of the cancer, what can we do to accelerate it? And now, fast forward, more than a decade of that type of thinking has produced many new FDA-approved drugs in a variety of ways, and I'll go through some of them. We'll talk about this later, but for patients with pancreatic cancer, we're behind other tumor types where these types of approaches are having more effect than they are for patients with pancreatic cancer, for reasons we can talk about. So one of the fundamental things to understand is that there was a shift with immune therapy in that the drugs that we are giving to patients are not toxins poisoning tumor cells or even small molecules interrupting tumor cells, but they actually often act on the immune system directly. So that was a big shift, right? These are drugs for the immune system, not anti-cancer. And it turns out when you turn on the immune system, then the immune system can find the cancer. So probably the real breakthrough, although there were a few efforts along the way, but the big breakthrough was immune checkpoint antibodies. These are monoclonal antibodies that a technology that had been developed in the 80s that were mostly developed against cancers, as I said, but then it was learned how to develop them for turning on the immune system. And the two types developed, they're against molecules that people probably have heard of, And certainly if you watch TV, you'll see all these drugs are now advertised: anti-PD-1, anti-PD-L1, anti-CTLA-4. There's multiple versions of these now FDA approved. Again, mostly for patients with other types of cancer. Those work by actually stopping the brakes of the immune system. So it's like a car on top of a hill. It will go down the hill if the brakes are off. So these are really molecular ways to allow the immune system to elaborate itself. And they really work in some cases amazingly well. Another major breakthrough shortly thereafter was the development of engineered cells, the patient's own cells, because it was realized that even giving drugs to a patient like I just talked about might not be enough. If we take them out of the body and engineer them with gene therapy and put them back in, we can point them toward the cancer cell in a more robust and potent way. And that those drugs were about 10 years or maybe 8 years ago or so started receiving FDA approval. Again, not for patients with pancreatic cancer, but it was a sign of hope. Patients with leukemia, patients with lymphoma. So those are cell therapies, and they're really individual. We take blood from a patient, engineer the cells, and give them back like a white blood cell transfusion. And a third type of immune therapy that maybe predates all of that, but has really yet to find its FDA-approved niche, are vaccines. So vaccines are funny because we think of childhood vaccines, and childhood vaccines are all about preventing, or the flu vaccine. You have to get the flu vaccine before you get infected with flu. But here we're trying to use that sort of stimulation, that sort of vaccine power to start an immune response in a patient who already has pancreatic cancer. And that's been very, very tough. And there's been many good ideas that look great in the laboratory that haven't panned out. I would say recently there's new excitement, and we can get into this, using mRNA technology, the very technology that was used for COVID vaccines now being applied and led to a very active, very promising clinical trial right now. So you have checkpoint antibodies, you have cell therapies, you have vaccines of all sorts of molecular formulations. It's almost, Andy, it's almost too much to keep up with right now. And that's why when we have conferences, the young investigators come pouring in, because it's super exciting technology, getting after very refined biology, and we've never been more hopeful.
Yeah, no, thanks. That's, I think, very helpful for us to all get oriented. And I think very important distinction that you made there is we shifting in this focus on cancer therapies from using toxins to target and kill the tumor cells to how do we boost the body's normal response to the tumor and leverage the immune system to make that change and to attack the tumor for us, which will hopefully be safer and more effective going forward. And you went through a couple of examples there, and I think Nobel Prize winning discovery of checkpoint inhibitors and bringing them into drugs, and near miraculous effects that has had in melanoma and other cancers. Incredible impact that engineered T cells have had in blood cancers. A lot of that work coming out of Penn, showing things forward. So, and you've alluded to a couple of times that pancreas cancer has been more of a challenge. It's a little bit tougher. Are there some reasons? Are there some features of pancreas cancer that make it harder to target?
Well, we've been spending a lot of time trying to understand that. And, pancreas cancer puts up a force field against the immune system. And we think that happens from the very early moments. And we had to get our head around the fact that the biology of pancreatic cancer is different than some of the other types of tumors where immune checkpoint, for example, works a lot better, like lung cancer or melanoma, where in those tumor types, there seems to be a very rapid response of the immune system in many patients, maybe even the majority of patients. And those T cells get to work, but they get exhausted and they need some reinvigoration. What we learned in patients with pancreatic cancer and in many of our model systems is that the T cells are just never activated. That first step toward recognizing the cancer doesn't happen. And there are many mechanisms we're revealing where the tumor cells, pancreatic tumor cells, in some ways in a unique way in cancer biology, put up this force field. They they surround themselves with other immune cells that are actually suppressive. They orchestrate a very fibrotic environment that is low on oxygen, very hostile, the last place an immune cell really wants to be and flourish. And we're learning why that happens. And one of the main drivers, we think, and of course we're still learning, is the oncogene KRAS that underlines the biology, the malignant biology of most patients with pancreatic cancer. I think your audience will be familiar with that, KRAS. And KRAS is an instigating oncogene. It's an ordinarily normal, very vital protein, but becomes mutated and doesn't turn itself off, and it just drives pancreas cells to grow and grow and invade. But what we've learned is it also sends signals to create this hostile environment, to bring in those immunosuppressive cells, to bring in immunosuppressive fibroblasts, to build this stroma, and that's one of the big challenges for patients with pancreatic cancer. We're facing the power of the KRAS oncogene, which sets up this shop of immunosuppression so early. So we've done the experiment, the clinical trial, where we give patients with pancreatic cancer checkpoint blockade, trying to turn on T cells, but there are no T cells to turn on, relatively speaking, so nothing happens. And those drugs are not approved for patients with pancreatic cancer, with one exception, and that's the microsatellite unstable high patients, MSI high. It's about 1% of patients with pancreatic cancer. Those patients will often commonly respond to the checkpoint antibodies. That is not, that's only 1% of patients, but trying to be an optimist about it, those patients do respond. So this notion that immunotherapy will never work for patients with pancreatic cancer, that's just not right. We have that example. So we have to be smarter about derailing these immunosuppressive pathways. And that's put us down a trajectory of therapeutic approaches that is different than what we've been doing in most patients with melanoma, renal cell, lung cancer. And we continue to learn so much about the challenge we're facing.
Yeah, no, thanks, Bob. And I think that answer also starts to encapsulate where I started, of why this research community is so special and where you are, that we can go from the old story of bad news of pancreas cancer is a wily tumor, and it finds ways to avoid all the treatments that we want to throw at it. But the research is being able to look and be hopeful and say, well, there's a glimmer of something that can work here. There's the things that we're learning about the biology that are going to give us hope that this will work in a broader group of patients in the future. So maybe that's a good way to segue to talk a little bit more about the future, what you're excited about, what you're working on now, and where things are going. So maybe, in the short way of asking is, what really gives you hope and what gets you excited about immunotherapies for pancreatic cancer right now?
Well, I have to say that, again, there are many new ideas that have made their way to clinical trials. And so what really gives me excitement is the opportunity to have patients enroll on those clinical trials, because we want to understand the best treatments as soon as we can. Biologically, we've begun to understand the power of vaccine approaches and starting an immune response. The recent data of building an mRNA vaccine and other types of formulations, not just mRNA, that target other mutations in pancreas cancer, sometimes patient-specific, targeting the KRAS oncogene, which, by the way, is mutated, so it's foreign, and the immune system can see it, actually. These are really exciting efforts. They're all early. They're not FDA approved, but some of them have advanced to randomized studies that could lead to FDA approval. I want to drill down a little bit more about KRAS. I talked a lot about it. So we've known it's a problem, and it's a driver, and it's a reason pancreas cancer forms. We have not been able to stop it with drugs until very recently. The most exciting thing that's happening in the field are new inhibitors using completely different chemistry that didn't exist a few years ago for orally available drugs, pills, that block KRAS oncogene and stop it in its tracks. In our laboratory models, when we give a variety of these agents to see if they can kill and stop the growth of pancreas cancer, it is by far and away the most effective approach we've ever seen in 15 years. And the KRAS inhibitors that are currently approved are relevant for only a small subset of patients with pancreatic cancer. But there are literally dozens of these agents coming down the pike. There's many clinical trials, and the early results are exciting. We have to learn about the side effects, of course. And so we took a look at the KRAS inhibitors immunologically. Is it possible that there's synergy? The short answer is it's amazing. So in the experiments, we see that the KRAS inhibitors don't work nearly as well, and relapse is much more common if we don't have an immune system also at play. On the other hand, if we activate the immune system at the same time, this is the laboratory, activate the immune system at the same time as we block KRAS with these highly effective drugs, we see what we've long been looking for, an instigation of an immune response that's very effective. We can even stop giving the KRAS inhibitor and just let the immune system take it from there, and we're seeing cure after cure after cure in the laboratory. We want to get that rapidly to patients. We haven't seen anything this exciting, and it was unexpected. We published this paper more than a year and a half ago and have other data since then, and it's been like, really? Like there wasn't a great uptake, but we're seeing now as more and more folks look into this that this is a reality. I'll say one final, unless we want to pause there, that because there's a lot to unpack with the KRAS inhibitors. I know that patients are aware. More than 90% of patients and families listening hear that their loved one has a KRAS mutation. So there's a huge demand to get on these clinical trials, and there's more patients than access right now. So we have to work through that. But it's very exciting, and there's reason for hope. It's not false hope. This is pretty exciting, and can't wait to see where it goes.
Yeah, no, that's great to hear, and it's good to hear. I think people have been waiting for, and now are very excited about these KRAS inhibitors coming online. I think as they move into the clinic, maybe we're learning about there are some challenges associated with them, including durability of effect. So in that sense, being proactive about looking at how to use them along with and alongside other types of therapies, and including immunotherapies, and as you started from, as KRAS is one of the big tricks that the tumor has up its sleeves to avoid detection of the immune system, is this going to be really something that begins also to unlock the power of immune therapies for pancreas cancer?
It could be, the prospect of KRAS inhibitors with immune therapy is something we think is very promising. But it's a theme, Andy, that you alluded to there. This is a really complex tumor. I think the ultimate type of therapy that will be most effective will be a combination therapy. We have to come at this from all fronts. So one of the advances, we don't often talk about this, but if you go back 15 years ago, we didn't even have a chemotherapy regimen that was reliable for patients initially diagnosed with, say, metastatic pancreas cancer. Now we do. They're not curative, except perhaps, there's a small subset of patients who get complete remission, but they're a backbone upon which we can build. I mean, and it continues to have some innovation there where there's multiple approaches. And now we're seeing, can you add immune therapies? Can you add KRAS inhibitors to the backbone of chemotherapy? Can you start with chemotherapy, then stop it, and come in with a maintenance therapy? So combinations are not just piling on. You can also have sequential combinations. That's really been an exciting way forward, using something called PARP inhibitors, which is for another subset of patients with pancreas cancer. So I just wanted to make that point. And when you do combination therapies, then think about the community of researchers you need to bring to bear. This is something Lustgarten Foundation does so extremely well, to bring clinical scientists and mathematicians and immunologists and cell biologists. Like, we're not going to solve this problem one lab at a time. It's going to take networks of people. It's what Lustgarten does so well.
And thank you for that, Bob. And it is something that we definitely try to make sure that we use our position as a representative of patients and their families to really push things forward as fast as possible for patients. And a big part of that is items you mentioned already: clinical trials, learning from the patients, collaboration, moving forward, breaking down barriers, whether they be with funders or drug companies, or even just finding ways to bring new drugs together in combinations to help to drive things forward. So maybe this is a good chance to give you the opportunity to talk about some of the work you've been leading on that I think covers a lot of those points. So I think, as I mentioned in the intro, you've been a main PI for a couple of very impactful clinical trials called Prince and Revolution. And as I look at this purple background, people might sense a theme here of how that that naming scheme came, which I'll leave it to you if you want to comment on that. But maybe, if you want to talk a little bit about, what was so special about those trials, why is it important to take a different approach? And, how do you use the learnings that are coming out of from the patients enrolled on those studies to inform next steps and help us to, not just continue to make progress, but to accelerate our progress?
Yeah, that's we've learned a lot through those trials. So I had the privilege of leading the trials you referred to. They have those names, Prince and Revolution. This was an effort that started initially with the Parker Institute for Cancer Immunotherapy, and at its heart was a consortium of 7 institutions across the country that worked together on this problem and worked incredibly well. The idea was, how can we bring immunotherapy to our patients? And there were laboratory observations that we had made that, as you mentioned at this top, this molecule CD40 can be used to activate the immune system. This is actually not cutting the brakes. This is a way to step on the gas. And we had data in the laboratory that agent, with or without checkpoint blockade and chemotherapy, could be effective. So we had done some early clinical trials, and we knew that this was a safe approach. And we launched this study called Prince because of the purple theme of the musician Prince, I suppose, and brought these groups together and ran a clinical trial of various combinations. And there were 3 arms to the study. It was eventually published, and folks can check out the results. We moved very quickly. The whole study was completed in a year and a half because of this incredible collaboration. And the idea was that the combination of immunology and chemotherapy could benefit patients. The study did not randomize patients to chemotherapy alone. And we heard from patients that was, that's an issue when they go on clinical trials. Are they going to get be randomized to just control? So we didn't do that. And what we learned is the power of collaboration and how patients joined, and we were able to collect many biosamples and have done a lot of science. We saw many patients with major regressions to their tumor, some of them quite durable. And we wanted to know, are we seeing that more commonly than we would have with chemotherapy? And when we looked into this, we found out that it was true, but just for a certain subset of patients, where we could identify them based on the health of their immune system, whether or not they had a certain number of cells in circulation compared to other patients. And if they did, their overall survival was twice as high as what we normally would expect. When you treat everybody, you start to lose that signal. So we learned 2 things. The first is that this sort of rapidly enrolling patients and quickly understanding how patients are doing so we can choose this therapy, that therapy, that therapy to go forward led to the next platform study, which was Revolution, which was a true platform study, which is sequential cohorts of adding new ideas to chemotherapy. So again, the same group of folks enrolling patients. We added checkpoint, we added autophagy inhibitors, we added combinations, we've added gene therapy approaches, and we're finding it a very rapid way to see, is this a good idea or not? And if it's not, we move on to the next best idea, and if it is, we expand the cohort. And it's happening now. It sort of moved beyond the Parker Institute, as you know, Lustgarten and other organizations involved, and it's been pretty exciting. The other thing, which is going back to what we learned in Prince, is this power of precision. Like, who are the patients that will benefit? So just as I said, one therapy or one lab's not going to solve this. I don't think we have a one-size-fits-all therapy for everybody. And I think we learned in Prince that there's, for whatever reason, a subset of patients that seem to respond to this chemoimmunotherapy. So we're now designing a trial, and of course, Lustgarten scientists have been heavily involved in thinking this through, where we identify those patients up front based on an immune test, and then treat them with the therapy that we've been working on to see if we can show power there. So, every trial, we learn from the trial, we learn from the patient samples, we refine our approach. And this used to take 5 years to turn that crank. Now it's taking, 5 months. And that's the kind of speed that we need. And we're looking for big effects. If we find something that changes survival by 2, 3, 4 weeks, we got to do better than that. So we're looking for major effects, and it's been really exciting and a novel approach, and it really allowing us to think about precision approaches. Precision meaning matching the right therapy with the right patient.
And I think, and staying on that point a little bit, Bob, so I think that's a theme that we've talked about a couple of times, right? Whether it be the MSI high patients, which represent a small fraction but benefit from checkpoint inhibitors alone, which you've shown from Prince and this immune markers that can be predictive that show people doing better. And when we talk about with KRAS, that there are different KRAS mutations and different specific KRAS drugs coming. But going back to what you saw in Prince, and at the risk of adding another file to your inbox or more to work on, it seems, more and more information coming up that the immune system might not only be a great target for attacking the tumor, but may also be a real sensitive sensor of what's happening in the body and when tumors are arising, or who's likely to respond to a drug, as you showed with Prince. So maybe, and this is maybe now getting a little bit more speculative, but I'm wondering if you could comment a little bit on, where else might we see the immune system playing a role in knowing who to give what therapy, knowing what therapy to give to which patient, knowing who's responding, even perhaps detecting tumors early. Is there a role for looking at the immune system as a biosensor of what's happening with the tumor?
Well, there absolutely is, and that's the job of the immune system. Mm-hmm. So the immune system does two things that the other systems of the body generally don't do. First of all, it goes everywhere. It's checking every cell out all the time. And the second thing is what it sees, it remembers. There's a memory about it, and this is super powerful. So there is a hypothesis that the first awareness that there's a tumor in the body is actually at the hands of the immune system. But are we listening? The immune system sitting there screaming, this is not good. And we're learning how it recognizes cancer. It alters the immune subsets. It starts a T-cell response against, say, mutant KRAS or other mutations. It forms an antibody. Could we use those signals to detect? Well, we've started something here at Penn called Immune Health, not immune therapy, Immune Health, exactly your point. Trying to look at 10,000 profiles of individuals to see if the immune system can be used for early detection number one. Number two, can it be used to fashion therapy? A bit like I described, we learned from Prince. You know, if a patient's immune health is in this flavor, maybe this is the right approach versus some other approach. And then there's also, once you start treating a patient, to monitor the immune system to see if it's working. Because the last thing we want to do is put a patient on a trial or on a therapy for 8, 10, 12 months when we have data that the immune system is not being activated. We need to do something else. So we constantly want to listen to the immune system. You know, this is not a new idea. People have known about this, but our ability to listen has completely changed. The technology is revolutionary. We can study, you know, 1 million parameters in 10 seconds. When I started, you had to look down a microscope, you know, one cell at a time. We'd never get there. And the ability now to use AI to help us listen is really an amazing opportunity. So that we need large data sets to begin to capitalize on all these opportunities that you point out. By the way, can I say one thing? This is not the first crazy idea to try to understand a patient's tumor or the patient themselves. You know, 25 years ago, someone said, hey, why don't we start sequencing tumors? And maybe that will tell us, right? So 25 years later, we're sequencing KRAS mutations. We're looking for MSI high. We're looking for BRCA1 and 2 mutations. Now we're saying, and back then when that started, people thought that was a crazy idea. And now it's routine. In fact, we're upset when a patient comes to us and that work hasn't been done yet because it's so vital. Now we're saying, let's take that strategy and extend to the immune system. So let's effectively sequence the immune system or study the immune system because of all these possibilities. As a sensor, it's telling us how to treat a patient. So it's an opportunity we have now because of past experiences, but also new technology and immunology is unbelievable.
Yeah, I think that's something we'll come back to at the end. As sort of what's on that wish list of tools you wish you had in the toolbox as a researcher. Let's come back. But you know, moving on it, and one, I'm just going to give a reminder to folks listening in that the Q&A box is open. I do see some questions coming in. We'll come back and address them. But if people have questions that come up, feel free to type them in as we discuss. But I want to, turn back to something you mentioned earlier, Bob, around vaccines, right? And how vaccines are, oftentimes an immune therapy that we don't think of as an immune therapy because it's something we're used to getting every year or taking our kids for routinely. But have really been showing, some promise in pancreatic cancer with a study that Vinod Balachandran ran out of Sloan Kettering. A lot of work that you've been involved with in your group and in collaboration with Liz Jaffe, who's our chief medical advisor for Lustgarten and also professor at Johns Hopkins. So I wonder if you could, maybe give a little bit more of sort of the role that vaccines can play, and both in kind of a general sense of, as a out of the box potential therapy for folks and what stage that might be used, and also in a kind of a hyper-personalized way as it's been looked at in a couple of instances.
Right. So happy to do that. Vaccines have been promised for cancer for 30 years and have not delivered the way we want. And I think we now have the knowledge of the immune system to craft this better. So why give a vaccine to a patient? First of all, it's a little bit of a misnomer because we usually think of vaccines as prevention. I want to get back to that in a minute. But we understand now that for a vast majority of patients with pancreatic cancer, and a little bit unique across patients with cancer, the immune system just doesn't get itself going to recognize the cancer. That's what the vaccine is for. Can we tell the immune system in that patient, it's time to wake up and focus your attention on the tumor? We've learned targets to wake up to. So we now know that pancreas cancers do harbor mutations that the immune system can see. We now know, and this is work that Lustgarten has helped fund in my own laboratory, we do know that the immune system can recognize the mutations in KRAS. Not every last patient, but it's certainly there. There are other non-mutated targets that such as something else we're working on that are not mutated. But none of these things are recognized unless you convince the immune system it's time to do that. And so that's why you need a formulation. We have the target, we just talked about that, but what's the formulation? Well, people have used all sorts of things, peptides, cells, but now we have the power of mRNA and other types of gene therapeutics, which seem to be pretty amazing. And they're not so hard to manufacture. And they can be made patient-specific. I mean, these are all pretty rapidly and pretty successfully. And these are some of the notions that have gone forward to a randomized phase 3. Okay, so where can we realistically expect vaccines to work? We have had 2 decades of experience for patients who have advanced pancreas cancer, who've been through chemotherapy or multiple rounds of chemotherapy. That is a really difficult immune system to wake up with vaccines. Some of the exciting work now is in, for example, patients who have earlier stages of pancreatic cancer, where the tumor has been resected, they've received adjuvant therapy, the chemotherapy and other types of therapies that you might give after surgery, and to prevent relapse, while the tumor might just be a few cells that they're trying to relapse, that's where we vaccinate patients, and that's where we're seeing great promise right now. So the immune system is just given a leg up by being used earlier on. So what's the implication of that? Why can't we use the immune system to wake up the immune system to pancreas cancer before the patient is a patient? This is getting back to where vaccines actually, we know they work, which is in prevention. Here we need to wake up a T-cell response, not necessarily an antibody response, and that's very hard. That's not usually done in childhood vaccines. So now we're seeing a shift to, can we vaccinate individuals who are healthy but at high risk of pancreatic cancer? And those trials, Andy, those have started. This is not Star Wars stuff anymore. That is happening. It's happening against KRAS. We have a trial against telomerase. It was just written about in the Wall Street Journal. The data is coming out from many centers. It's pretty exciting. Sometimes we call it prevention. Sometimes we call it interception, which is assuming a patient might have a very early lesion that we want to get rid of, a pre-malignant lesion. So just to say that again in other words, to take our clinical trial here, we are actually meeting patients who are not patients, they're healthy. They carry germline mutations in BRCA1 and 2, and we're vaccinating them with a DNA formulation to prevent the development of all sorts of cancers, including pancreatic cancer, which we have no other ways of preventing that in those individuals. So we're going to study very hard, and everyone involved in this work across the country is trying to understand if we now try to wake up the immune system in a healthy individual, what does that look like? Maybe it's logarithmically better. Maybe there are side effects that says we got to be careful. Huge opportunity to attack pancreas cancer before the patient is a patient.
Yeah, and totally agree. I think that is a very exciting place to wake up.
One thing I meant to say, none of that would have been possible had it not been for all the work in cancer immunology that has happened. Like, we are taking advantage of insights that we didn't have 5, 10, 15 years ago.
Yeah, and that's exactly where I was going. We're truly standing on the shoulders of giants in all the work that's gone in to know who is at risk for pancreas cancer. What does risk mean? What do early stages look like? What are the mutations that happen early? All of that real fundamental understanding of the disease and the patient and the biology is what makes it possible to do something that seems so simple, give somebody a vaccine when they're still healthy, right? But it's built on decades of
understanding who's at risk genetically and for other reasons, which is not work done by immunologists but other geneticists. So that is critical, as you said. We have that now.
Yeah. All right, so I'm going to switch gears a little bit because we've gotten some great questions coming in the chat. Some I think that will be near and dear to your heart for things that you're thinking about, and maybe we can kind of go through these a little bit quick of the lightning round level of answer. So
Okay,
the
good.
first question that came in, which I promise you wasn't planted, was around oncolytic viruses. What's going to be the role for oncolytic viruses in pancreas cancer?
That's still a maybe. Um, uh, it's possible. We got to get. I think the viruses are still being optimized. Those have great potential to activate the immune system. I think there's a lot of refinements going on, and whether we're going to end up giving it systemically or perhaps directly into the pancreas. There's a lot of work to be done, but it's still quite promising.
People who have known mutations or alterations, we talked about MSI, alluded a little bit to PARP inhibitors for people with BRCA mutations. How does having a known mutation in a pathway like in BRCA affect how you would think about immunotherapy for that patient?
Oh, it's we're still working on that. We don't know. I mean, I would just say you're going to read places where everyone, oh, we know. I don't think we know. There is, we're starting to get some empiric data. For example, combining checkpoint immune therapy with PARP inhibitors. A study done by my colleague Kim Rice was pretty eye-opening about that. Whether or not individuals with those particular mutations are more responsive to checkpoint. We know that that's true. That's the whole reason patients who are MSI high respond better. But not every patient with MSI high tumors respond to checkpoint. So there's a breakdown in our knowledge there. But the general concept that, and this is true in breast cancer and lung cancer, that not every patient is going to be responsive to every immune therapy approach. That's definitely true. We need more refined understanding of that. And right now, it's really important for us to know both germline, it's like such the things we just talked about, but also somatic, the actual sequencing of the tumor. Both of those are giving us immune information that's critical as we develop new therapies.
Staying on the mutation theme here, but now going to somatic mutations. A lot of questions about different KRAS mutations. My family member has a Q61. Somebody has a G12V. How much difference does the specific mutation in KRAS have on how you would think about treating the tumor generally right now, or with immunotherapy specifically?
Oh, a lot. That's a huge variable. We even know clinically the type of mutation can have different manifestations. So let's just take a few things at a time. So KRAS inhibitors. There are some KRAS inhibitors that are specific only for a particular mutation, such as KRAS G12C. They're FDA approved for patients with lung cancer. That is only going to be relevant to a very small subset of patients with pancreas cancer. There are other inhibitors that'll be specific for KRAS G12D, much more commonly found in our patients. But there are now KRAS inhibitors that are specific, and they're multi-selective, so they'll hit multiple mutations. Then the question is, well, which ones hit which mutations more potently? And all that work is evolving right now. On the immune side, this is actually very complicated. Different mutations. I said KRAS mutations can be seen by the immune system. Well, not every mutation has that feature, and not every human has an immune system that can be recognized in that way. And so you start thinking of combinations like this mutation with this type of HLA type, which is the white blood cell type, like a red blood cell, but for white cells. And, there has to be that combination. And when we find a particular one that's really good, we're disappointed to learn that maybe that's only 5% of all patients. Maybe it's only 8% of all patients. And the idea is, can we find 10 of those so that we can have... So we're kind of getting to the point where we're developing the medicine cabinet, and now we got to look at the patient. What kind of KRAS mutation do you have? What kind of HLA type do you have? What's the stage of the tumor? Other mutations, and craft a therapy specific for that patient.
Thanks. And there's a lot of great questions coming in. We're not going to be able to get to absolutely all of them, but I think there's one here that's really pertinent, and I'm going to use that to segue into my last question for you, Bob. And the question is really about how do we make this all go faster? How do we really accelerate the development of new drugs? And that's a big open-ended question. So I'll narrow it down a little bit for you, Bob. Let us know what's on your wish list. What do you wish that you could have or wish that you could see that's going to help you in the lab, you in the clinic, you in designing trials to push things forward?
Yeah, and so I think about this a lot as a cancer center director as we try to mobilize our own forces or collaborate with other cancer centers. So, you know, what's on the wish list? So, you know, we see that actual basic research is delivering the insights we need, whether it's genetics or immunology or cell biology. So that activity needs to be supported and flourished, and we have to be willing to take risks on people, particularly young scientists, who have quote unquote crazy ideas. I love it when someone says, "Oh, that's a crazy idea." Then I gravitate toward that. Like, what? Why is it so crazy? We have to have funding mechanisms for that, and we have to be willing to take chances. And this is, I think, what Linda said at the start. You know, high reward, high risk sometimes. So we need partners who are willing to do that. Once we have an idea and we're trying to translate, that also needs infrastructure and funding. The clinical trial apparatus that we use, particularly from industry right now, is slow and it's inefficient, it's costly, and we're not getting there. And if we could challenge our advocates and other folks who support us to join us in seeking a better way to do clinical trials. That's what we tried to do with Prince. That was one of the underlying lessons. Like, I think we found a better way, but it's costly. And you know, that is like, we have all these KRAS inhibitors. There's dozens of them. How can we most efficiently find out which ones are the best and get them to our patients without doing them one by one in a silo? I think that's a challenge for the country. I'd love the NCI to help lead that. And I know they want to, and partner with the companies to get these patients faster. And then I'd say one final thing, Andy, which is this is a long road. So we need to be resilient in our approach. We need to have enduring support. We need to be willing to make mistakes and learn and move forward. And I think we need to understand that this is going to take time and continued support, right? So we're not just going to rush in, figure it out, and leave. There's going to be a ton. I feel every time I meet a patient and get those calls from a patient saying, you know, why is this therapy not ready for me? It's just agonizing to think if that was my dad or my brother, and we're motivated by that to move as fast as possible, and things are moving faster. We are sharing data. There is collaboration. There's tons of consortiums, and the biology is delivering. We just need to double down on that.
Well, thank you so much for being with us, Bob. I think this has been incredibly illuminating for me. I hope it was for the audience, and I think you closed it out in the perfect way of really what's been the founding principles and motivation for LustGarten since 1998, that patients can't wait. We're moving with a sense of urgency, but we also know that we're here for the long run until the job is done, and that we're not gonna be limited by the way things have been done or the models that have been put in place, that we're gonna find the right tool for the job to push it forward. And you've been a big part of that, working with the foundation, working on partner things, and we, you know, thank you for that, and thank you for being part of this webinar today.
No pleasure. Thanks, Andy.
Thank you, Dr. von der Heide. You know, I really liked what you were saying about resilience, and we expect a lot from our pancreatic cancer community. We expect them to be resilient, and I'm always so proud of so many of our community members who really dig in, to support the community after they've lost someone, and of course, patients who really dig in to find the best therapy for their cancer. And I also really liked what you said about learn fast and fail fast. And I think that's the mantra of what the Lustgarten Foundation wants to do, so we can get therapies to our patients faster, and so that as a community we can really learn from patients and hope that one day patients will have longer survival rates. And we're so grateful for your dedication to your research and to our patients. And I know how much you care, and we're really so appreciative. So, and thank you for doing this today. Thank you.
My pleasure. Thanks for all that you guys do at Lustgarten and all your supporters. It makes a real difference.
Thanks, Bob. So thanks to all of you who joined us today and for really wanting to understand why immunotherapies are and will be so important to patients. Our next Lustgarten Live will be next month, and it will highlight the unique collaboration and impact that 3 of the Lustgarten dedicated labs are having on research. We'll hear from researchers from Dana-Farber, from MIT, and from the Salk Institute. Andy, this gathering of lab leaders promises to be a fascinating discussion. So please watch your emails and our newsletters for details coming soon, and we'll see everyone next month. Dr. Rakeman, thank you very much for hosting this very enlightening discussion. We really appreciate all you contribute to our program. So thanks, everybody. See you next month.